Hello, everyone, and welcome to another lecture on Tink Talks. We'll be looking at practical applications of immunology, and in the textbook that I use with my college students, this is Chapter 18. So let's get started. So we'll look at some terms and discuss a bit of history about vaccines and vaccinations. But before we get into vaccination, we need to look at this practice that was called variolation. And variolation is actually quite an old practice, dates back centuries. The Chinese used it, and we have record of it, where they would use this practice of variolation, in which the process that was used in Western culture in the 18th century, where they would inoculate smallpox into the skin of people. So they would take some scrapings from smallpox lesions on people that had smallpox. They would dry them out, and then they would scrape the residue into a healthy person. And as a result of it, they developed immunity against smallpox. However, it was dangerous because the mortality rate was significant. It was significant enough that it would be concerning, obviously, for somebody to have this done. But smallpox was such a contagious and mortal disease that people were willing to have this process done. They took their chances, and in many cases, it worked out well for them. However, the mortality rate was still high enough that a number of people died from it, even after having variolation done. So it was risky, but it conferred immunity against smallpox, and it increased your chances of not getting it. I mean, the mortality rate for smallpox was very high, around 30% or more. The mortality rate after having variolation done was only around 1%. So it was still a significant number of people that actually died from variolation, but it was not nearly as many as those who died from actually having smallpox itself. As I said, we have records of the Chinese using this as early as the 11th century, where we have record of a monk who would take smallpox lesions from people and the residue from it. This monk then would dry it out, put it in a mortar and pestle, grind it up into a powder, put the powder on the end of a hollow tube. I guess then it must have been a hollow plant tube, and then insert that up a person's nose and then blow the powder through the tube up into the person's nose, and that was their form of variolation. Later, as you can see in Western culture, that wasn't done. It was scraped into the skin. Vaccination is inoculation of cowpox into the skin, and it comes from the Latin word vacca or vacca, for cow. And we call it then vaccination because it all started with cowpox and how Edward Jenner used it in 1796. So this was quite a few years, even before the germ theory of disease had been developed in 1876. So we're looking at about 80 years before we even had any idea about germs and bacteria and viruses spreading disease and disseminating themselves in a population. Jenner notices that the milking maids for cows were developing cowpox, but they did not develop the deadly smallpox. And so he took note of this, and he interviewed many milking maids, and he found out that that was the case with many of them. Yes, I've had cowpox, but I've never had smallpox. And so what Jenner thought was, maybe this is something we could try to do ourselves artificially. Take this matter into our own hands rather than allowing people to get cowpox naturally and then develop immunity against smallpox. Why don't we just give people cowpox so that then they develop immunity against smallpox? So his first patient was James Phipps, and James was the son of his gardener, the son of Dr. Jenner's gardener, and he intentionally inoculated him with cowpox. And James became mildly ill from cowpox, and he recovered from it. And then a few weeks later, he intentionally inoculated him with smallpox. And little Mr. Phipps did not develop smallpox. This then was carried out on many people then. It was very successful. It was met with a lot of, in terms of Jenner and how he carried it out, it was successful. However, in terms of the general public and the officials making this something that would be approved and to be used on the population, that took some work on Jenner's part and others who supported this. But eventually, variolation was outlawed. You could say it was no longer allowed to be used, and vaccination then replaced it. And Jenner's his experiments and the methods that he used and the system that he used in order to prove this was very successful. And so vaccinations then came on the scene, and it started with Edward Jenner back in 1796. So what is a vaccine? A vaccine is a suspension of organisms, or fractions of them, and we'll look at that in a couple of slides here, used to induce immunity within a person or within a population if it's given on a mass scale. Herd immunity will result when most of a population is immune to a disease. So, for example, in the United States, measles was declared eliminated in the year 2000. And that would coincide then with herd immunity in the United States. And the reason why we achieved herd immunity was because of the incredible success of the measles vaccination, the MMR trio, measles, mumps, and rubella. And as a result of the administration, the mass administration of the measles vaccine, the number of cases plummeted precipitously. It was like falling off of a cliff, the number of measles cases in the United States. As the number of vaccines administered increased, the number of measles cases decreased greatly. It was a very strong, powerful correlation between the two of them. And there's a video link there, Eradication of Smallpox. It's a good one on TED Ed. So some principal vaccines that are used in the United States. DTaP. So we have, notice how the lettering of it is. You have a capital D, a capital T, a lowercase a, and a capital P. Well, the D and the T, the D is diphtheria, the T is tetanus, and then the A in front of the P is for acellular pertussis. So how is this concoction made up? So the diphtheria part is a purified diphtheria toxoid. And if a word ends in oid, it means it's like the prefix. So with the suffix of oid, this is like the diphtheria toxin. And the same with tetanus is purified tetanus toxoid. It's like the tetanus toxin, but it is not. It's a neutralized toxin. Obviously, you're not going to give the toxin because that could create major problems. So it's a neutralized toxin of those two in particular in this instance, diphtheria and tetanus. Pertussis is given as acellular fragments of Bordetella pertussis. So these are bits and pieces, polysaccharides mainly, and proteins from the cell wall. And these fragments are put together. The cells are fragmented, they're chopped up, they're fractionated, and then those fragments are put into this preparation. And then you may have heard of DTaP. So we have the same letters, but just rearranged. And DTaP for tetanus, diphtheria, and acellular pertussis is given as a booster after age 11. What's the difference between DTaP and DTaP? Well, there is less diphtheria and pertussis toxoids in there. So it's changed slightly, and it's given as a booster after age 11. Now, years ago, not all that long ago, I should say probably within 10 years, it was just given as the TD booster, the tetanus and diphtheria booster. But with the increase of pertussis cases within the past 10 to 15 years in the United States, it was decided that acellular pertussis needed to be added to this as well in order to reduce the number of pertussis cases. So now we have, instead of just the TD booster, it's the DTaP booster. Meningococcal meningitis, that's the purified polysaccharide from Neisseria meningitidis. And so this is, you have the carbohydrate portion of this bacterium, Neisseria meningitidis, and you take that purified polysaccharide carbohydrate, and that's what's used in that particular preparation. Haemophilus influenzae type B meningitis, polysaccharides conjugated with a protein. So here we don't have just the polysaccharide like with the meningococcal meningitis preparation. Now it's conjugated with a protein. And we'll talk about why there are conjugations in a few minutes. Pneumococcal conjugate vaccines, Streptococcus pneumoniae, antigens conjugated with a protein. So again, we have parts conjugated or joined with a protein. These are just various preparations for these. What are some other ones that are used in the United States for polio? We have the inactivated virus, the IPV, polio vaccination. Rabies is an inactivated virus. Hep A, inactivated. Influenza, and so on. And you can look at this list and you'll see these terms, what inactivated and attenuated. Okay, so inactivated means we could say killed, but technically the virus, I guess, biologically speaking, can't be killed, so we use the word inactivated. We say that's inactive. So this would be a, quote, killed virus, a virus that cannot replicate, that cannot get into our cell and make more of themselves. So we have the whole virus, but it's inactivated. As opposed to the word attenuated. Attenuated means to weaken. And so these are weakened viruses that are given. So in some cases, like with influenza, depending on what the strain is predicted to be that particular year, it'll be an inactivated flu virus or an attenuated one. Measles, mumps, and rubella, MMR, they're all attenuated as well. Chickenpox is attenuated, and Hep B, you get antigenic fragments there. So the closer we can get a vaccine to the real thing, the better the immune response. That's generally speaking, not every case, but generally speaking, the closer we can get to the real thing, the better and stronger the immune response, which means then, as a result, your immunity then has been boosted, and it will be a better immunity, and in some cases will last throughout your entire lives. For example, the MMR vaccination, those attenuated viruses, they act as natural boosters to us for the rest of our lives. So that in many cases, up to 95% of the cases, you will not get measles again. So it acts as a natural booster for us all of our lives. Whereas other ones, if we get further away from the actual virus or the actual bacterium, like with tetanus, we need boosters because it's a toxoid. It's not like tetanus. It's not the cell. We're not given tetanus. We're given something that is like the tetanus toxin. And so we're getting further away from the actual antigen or pathogen, I guess we should say. And so the further away you get from it, that will often lead to the need for more boosters later, periodically through a person's life. The effectiveness of vaccines, well, what do? They produce a primary immune response. So you have that primary response, the one that's slow but steady, which then leads to production of antibodies and long-term memory cells. So you're given in a vaccine something obviously that is not going to give you the full-blown disease or illness, but it's going to enable you to produce antibodies sufficiently that when you do actually encounter the pathogen later in life, you will have those memory cells that will become plasma cells so quickly and start dumping boatloads of antibodies into the bloodstream. And so when you have those second and subsequent exposures to these particular pathogens, you don't go through the primary response and all the effects associated with it, with illness and disease and so forth. So that is how vaccines primarily work. You get a primary immune response. And in some cases, when you do get a vaccination, it kind of wears you down because your immune system is working. It's working hard to develop a whole bunch of memory cells and plasma cells producing antibodies. But then once that, quote, artificial infection, once that clears up, everything's back to normal. But those memory cells remain. And so then, and now that's the key, that's the secret to these. Memory cells are made, and whenever a secondary response occurs, it occurs way faster. And you get so many more antibodies produced as a result of it. So you have a rapid, intense secondary response. And then the pathogen never gets a chance to produce its toxins, to get into our cells and cause all kinds of cytopathic effects. It never gets a chance to because we produce so many antibodies so quickly and rapidly, and then the macrophages take care of them so fast as well, and other immune cells, that we don't get sick. Vaccines generally aren't as effective against bacteria because bacteria possess polysaccharides. Viruses have a protein coat. And, well, what's the significance? Antibodies don't respond nearly as well to polysaccharides as they do to proteins. And so remember when I said they conjugate these polysaccharides to proteins? That's why. They take the polysaccharide, which generates a so-so immune response, join it to a protein, chemically bond them together, and now you get a very effective immune response. And so we conjugate these polysaccharides with proteins. Adjuvants are compounds. Those, and it's a number of different compounds. Aluminum salts are the ones that are used most frequently today. So some of them are inorganic compounds and some of them are organic compounds that are added to vaccines. It produces a stronger and more intense immune response. You get more antibodies generated, longer lasting immunity, and so one dose or fewer doses, at least anyway, is the benefit from adding these adjuvants to these vaccines. So these are compounds outside of the actual pathogen itself that are being artificially added to the vaccine, to the preparation of it, to get a better immune response. These are the different types that I alluded to earlier: attenuated, living, but weakened microbes, like the MMR vaccination. So because of them replicating and acting as a natural booster, we get lifelong immunity. About 95% of the cases end up with lifelong immunity. These are mutated microbes harvested from an artificial culture, so they can't create disease. They're not nearly as virulent as they would be if they weren't the mutated microbes that we get from these cultures. They don't recommend them for immunosuppressed or pregnant women because they can back mutate, and there have been some instances where back mutation has occurred, and it's rare, but it does occur. And if you are immunosuppressed or you're pregnant, it could affect the fetus, or it could affect the immunosuppressed person. So it's not recommended for those. So what do you do as an alternative? Well, we have these other ones that are not living but weakened. They are inactivated whole agent, or as I said earlier, killed, but we tend to use the word inactivated because when we're talking about viruses, we don't want to use the word killed, I guess. But anyway, for all intents and purposes, killed microbes, killed bacteria, killed viruses. So you have the whole unit, but it can't do anything. Notice whole agent. It's the whole thing. It's not bits and pieces of it that have been fragmented up. Toxoids are inactivated toxins that generate an immune response. The big ones are tetanus and diphtheria. And you have a series of injections, and about every decade you get boosters. And like I said, we're getting further and further away from the real pathogen, from the real bacterium or virus, and so therefore boosters are necessary because immunity will wane as you get further and further away from the real thing, from the whole agent. A subunit is a fragment that best stimulate an immune response. They're safer because there's no chance of them ever reproducing or ever back mutating because they're just bits and pieces, microbe fragments that sometimes will be conjugated. So we'll take those fragments, we'll join them with a protein, and as a result, you get a better immune response, a stronger immune response. And so the main reason why these were developed, these conjugated vaccines, were to deal with the poor immune response of infants, because we want to get individuals immunized as early as possible, because the longer you wait, the more vulnerability there is. So you want to be able to administer the vaccines as early as possible, but not too early, because of course that could be detrimental to the infant, but as early enough as possible, because that infant is vulnerable. There's a period of vulnerability when an infant just can't be vaccinated because of the limitations that we have and because of their weak and undeveloped immune system. So that's part of the main reason why conjugated vaccines came about. So that we can vaccinate people as early in their lives as possible, so that time of vulnerability gets less and less and less, to the point where hopefully one day it could be an administration right at birth. rather than waiting a few months to give these vaccines in some cases. Nucleic acid vaccines, those are plasmids of DNA. Plasmids are circular pieces of DNA from bacteria, and we inject these plasmids into muscle. And they'll stimulate the production of proteins encoded in the DNA. So the plasmid will incorporate itself into the DNA of the muscle cell. Then the muscle cell, as it divides, it will produce this bacterial DNA. These proteins then that are non-self proteins, they're not part of you, they're part of the bacterial cell. When those proteins get produced within our cells, they're going to stimulate an immune response. And so as a result of that, you have this plasmid incorporated in your DNA, and every time your muscle cell divides, it's like the lysogenic cycle, this protein gets produced as well. Now, muscle cells don't divide very rapidly, but it creates an immune response that continues as the cells divide. And also, a gene gun is used in the administration of nucleic acid vaccine, which means no needles. And also, there's no refrigeration needed for these, which is a big deal, because if we want to vaccinate people who live in remote areas, in remote villages with no electricity, no chance of refrigeration, this is a great alternative to that. So there's no needles, there's no sticking of people in places where bloodborne diseases are rampant. We have no need for needles and the exposure of these pathogens outside of the individuals. And also, like I said, if there's no electricity, no big deal. We got a gene gun, and the gene gun can deliver these plasmids, and there's no need to refrigerate these plasmids. So it's a great alternative. More research and development needs to be put into this with nucleic acid vaccines. Now, speaking of research and development, vaccine research and development has declined because of these primary issues. Popularity of antibiotics. Antibiotics are used to death. We are using them far too much for far too many illnesses. We get them prescribed far too quickly. Some doctors are willing to give them out far too easily as well. So we are just taking way too many antibiotics, and of course it's led to antibiotic resistance on a global scale. But antibiotics are so popular, they make so much money for pharmaceutical companies. It's an incredible amount of money that's made from antibiotics, and so they're not going to put as much money into vaccine research and development because they've got the antibiotic money train moving right along. Fear of litigation, and there is a serious fear of litigation. Many lawsuits, multi-million-dollar lawsuits, have come about as a result of poor vaccine development and research. People die in these, and they test these vaccines for so long, but yet still there are a few individuals that unfortunately develop some kind of problem, and it's usually due to their genetics. And so there are many ongoing, even now ongoing lawsuits against vaccines. Lack of profitability. Again, the pharmaceutical companies are looking at the bottom line, how much money can be generated from the production of these drugs. Vaccines just don't generate a huge amount of profitability. The amount of money that goes into developing a vaccine is astronomical. It's anywhere, I've seen from $50 million up to, it's a wildly wide range. It's $50 million up to $1 billion to develop an effective vaccine. And the time it takes, anywhere from about 10 to 15 years to develop a vaccine. It costs pharmaceutical companies an incredible amount of money, and it takes a long period of time. And so as a result of this lack of profitability, these pharmaceutical companies, their businesses, they're not going to put their research and development dollars into vaccines, especially if it's if it's a single administration or just a couple of administrations and then you're done, like measles, mumps, and rubella, that's it. We got it when we were young, and that's no more boosters. It's over. They're not making any more money off of us because it's, with vaccines in some cases, it's a single or just a couple administrations that are given to an individual, and then that's it. We are no longer a commodity. And it seems like we're in a depressing situation, but that's kind of the fact of the matter and the reality of this situation. The lack of profitability preventing vaccine research and development, which we desperately need, especially what's going on now with SARS-CoV-2. We've got to develop a vaccine against it. We can't wait 10 to 15 years. I mean, this vaccine development has to be put on the fast track. And we'll see what happens with the development of this vaccination, if it ever does happen. Let's hope it does. Safety issues. Developing the disease itself, which it does in some cases. People develop the disease. You're getting a vaccination against the particular disease, but ironically, you then and unfortunately develop the disease. There was this link between measles, mumps, rubella vaccine, and autism. Mercury was thought to be the culprit. It was an adjuvant in MMR, but it's no longer used in vaccinations, so it can't be the culprit and hasn't been used for decades. And the finding that this British pediatrician that he made, that he said that there was this link between MMR and autism, was found to be false. And yet people are still perpetuating this false information that measles, mumps, and rubella leads to the development of autism within children. And it was proven and found to be false, that is not the case. So what are we looking at with the perfect vaccine? One that would be affordable, and it is. I mean, the measles vaccination is only about, right now they're delivering it to Africans, people in Africa, for less than $1. It's around 70 cents a dose. So it's very affordable. We need one that's heat stable, that doesn't need to be refrigerated, can be orally administered, not through a needle, multiple antigen, so to be able to deliver a number of them at once so that we don't have to keep coming back again to we don't have to keep going to the doctor, or if you're working at a clinic in a developing nation, you don't have to keep going back to a village and administering these antigens again and again. And a single administration given at birth, that's the perfect vaccine. So that, and delivering it at birth so that there isn't that period of vulnerability after birth. Will we get there? I don't know. I mean, we have these roadblocks that are preventing us from getting there, but hopefully some of them can be moved out of the way, and we can develop this perfect vaccine one day. There are so many variables that come into play: political, financial, legal. There's so many issues. It's an incredibly difficult process to develop a vaccine. It's so expensive. There's so many politics involved, lots of government red tape, and tons of money that are involved in developing vaccines. So, monoclonal antibodies, and this is the last topic I'll talk about in this chapter. So, what this graphic displays here, I'll run you through it. It makes sense when you look at it, rather than me just rambling on about it point by point. So, we start up there in the upper left. We have a mouse. Always a mouse, the poor mice, immunized with antigen X, whatever that happens to be. And the mouse spleen produces plasma cells that secrete antibodies against the antigen. So remember, plasma cells, that's down the line. So you have the B cells that then differentiate. They go through clonal selection and expansion and proliferation, and they make loads of these memory cells. The memory cells then produce the plasma cells then produce the antibodies. Okay? So, at the same time, we have myeloma cells that are unable to produce antibodies, or HGPRT. It's an enzyme. But we're not going to concern ourselves with HGPRT. We're going to concern ourselves with the antibodies. These are myeloma cells, are cancerous B cells. These are B cells that have been induced to become cancerous because we want the rapid mitosis. We want rapid cell division. So we did step one. We immunized mouse. Mouse is busy producing antibodies, nibbling on his food, enjoying his life while he's producing these antibodies. At the same time, we have cultured these myeloma B cells. Then comes curtains for the mouse. The mouse spleen is removed, and the plasma cells from the spleen are isolated and mixed with myeloma cells, the cancerous B cells. Cell fusion is induced to produce what are called hybridomas, and it comes from hybrids, a combination. So we take these cells that can reproduce very, very rapidly, and we combine them with the B cells, the plasma cells from the mouse, and we get what we call a hybridoma. The hybridomas then we have the best of both worlds. Rapidly producing cell, and it produces the antibodies that are desired. The unfused plasma cell dies, the unfused myeloma cell dies, and the hybridomas are selected, and then they are grown in bulk, and we get loads of them. And loads of hybridomas are going to produce loads of antibodies. And that's, again, this is all called, these are abbreviated MABs, or monoclonal antibodies. So how do we apply them? What's the use of a MAB? They're used, first of all, like I said there when I was describing the diagram, used because B cells, which produce potent antibodies, reproduce only a few times. Their reproduction rate is slow, and they don't produce that many times. So we take normal B cells, we combine them with cultured myeloma cells, which are reproducing uncontrollably and rapidly. Therefore, as a result of that, we get large amounts of antibodies. So we have fast reproducing cells that are able to churn out loads of antibodies. We're going to use MABs for pathogen recognition. The biggest application, though, are pregnancy tests, most definitely. That is the most commonly used application for MABs. Suppression of tissue rejection and tissue transplants. Treatment of illnesses. Possible cancer treatment, because we're using the hybridomas or the myeloma cells combining together with normal plasma cells. There are rejection issues because the antibodies are of mouse origin, and so some people will reject them because of the difference between, obviously, humans and mice. And there are a few MABs that possess mostly human antibodies, less rejection issues, but they're very costly because you're getting these antibodies from people, from humans, and so this is going to be a little more costly than getting them from mice. There's a video there, a link for a pregnancy test. That's a TED-Ed video. If you want to go to TED-Ed, search for pregnancy tests, and it's a good video on how they work. So let's look at that. So with this pregnancy test here, you have the control windows, and they're showing you, okay, this is what it looks like if it's a positive test. You get the red area. The test windows are below that in this particular pregnancy test here. You'll see lines that form, dots that form. It just depends on the pregnancy test that you actually choose to use. But there's always as far as I know, and I don't use pregnancy tests very often, by the way, but as far as I know, the color red is always used. So what you have here, if you look at this little diagram on the left, the free monoclonal antibody specific for HCG, human chorionic gonadotropin. And so this one is a free one, meaning it's in that test window area, that bubble. And with it carries an enzyme that will create a chemical reaction that will produce the color red if HCG is present. Then, if you look down at the bottom there, number 2, there's a capture MAB that's bound to a substrate. So there's part in that test window where all of these capture mAbs are stuck. They're like rooted into a substrate. They're not moving around. They're not free like the one that's carrying the enzyme that lead to the production of the red dye. Then, when you urinate on the pregnancy test and you are producing HCG, which will come out in your urine, the HCG will travel through and be absorbed by the cotton in the pregnancy test. And if HCG is present, the HCG will be picked up by the free mAb, the one that's floating around, and they will then join to the capture mAb, the one that's rooted into the substrate, and they will form a sandwich. And as a result of this, a chemical reaction occurs through the use of enzymes, those magical proteins, and it creates a color change. If there is no HCG present, there will be there will be no color change, obviously, and there will be no sandwich that forms because the HCG is not there, so there's nothing for the capture mAbs to bind to, and there's nothing for the free mAbs to bind to either. And so they'll just the free ones will just continue freely floating around in there until HCG is present. Now, the HCG has to be at a certain concentration. So you may be pregnant, but your HCG levels are very low because it's so early in your pregnancy that you're just not producing it at a concentration high enough. So you wait a few days, or the advice, of course, is to urinate early in the morning because that's when the amount of HCG is going to be the most concentrated. So anyway, just you can watch that video on TED-Ed, and it explains it well and has some pretty cool animations with it too. So that's it for this discussion of Chapter 18. I hope you enjoyed this lecture. I hope it enlightened you on vaccines and vaccinations. There's so much more to say about vaccines, but that's sufficient for this particular discussion here at an introductory level. So if you like this video, which I hope you did, hopefully it helped you understand these topics. Check out my other videos as well. Share these videos if you so desire to do so, and be sure to smash that like button. Thank you for watching.